For a long time, anti-greying was mostly myths, dyes, and vague hair vitamins. The problem is that true age-related greying unfolds over decades, so proving that anything slows it down in humans is slow and expensive. But over time, we've learned much more about why hair loses its colour, which lets us look for plausible candidates instead of guessing. One of the most promising directions is targeted antioxidants, molecules that help the follicle cope with oxidative stress before damage becomes permanent. Among them is luteolin, a plant flavonoid already used in dermatology, which has now been tested directly in a greying model. Before we can see how luteolin might help, we need to understand how greying is triggered by oxidative stress. Paint for hair comes from deeper down in the bulb, where mature pigment cells load melanin into the growing hair. Higher up is the bulge, where pigment stem cells live. They replace worn-out pigment cells over time, supplying new ones to the bulb. Like any active tissue, this system runs on energy chemistry. As cells burn fuel, they release tiny reactive byproducts, a bit like sparks from a fire. Oxidative stress is what happens when there are too many of these chemical sparks and not enough defences to clear them. In small amounts, they act as useful signals. But when they build up, they start to damage DNA, proteins and cell membranes. Inside the follicle, that stress can weaken the stem cell bank in the bulge, damage the pigment workers and the enzymes they use in the bulb, and disturb the signals that link both parts together. Over time, those changes push the system toward greying. With this in mind, scientists wanted to test whether antioxidants could really slow greying. To do that, they turned to mice. Standard mice are not great models for human greying. They rarely show the gradual greying pattern we see in people, and even if they did, waiting for it would take too long. So a team at Nagoya University used special mice called RET-Mice. These are genetically altered mutants with a RET gene that makes their hair follicles cycle much faster. It's like pressing fast forward on aging. They start with normal dark fur and then develop grey hairs in a way that mimics human greying. The team followed both normal mice and RET mice over time, counting how many hairs were dark or grey at different ages and looking inside their follicles. Then they added luteolin. Some mice received a 1% luteolin solution applied to the skin, while others received luteolin mixed into their food. In both setups, treated mice went grey more slowly and retained more pigment stem cells and pigment-producing cells than untreated controls. In this lab model, greying starts when support cells in the follicle age get stressed and stop sending good survival signals. Pigment stem cells then fade away, fewer pigment cells reach the bulb, and more hairs grow in grey. With luteolin, that slide slowed down. Support cells looked younger, oxidative stress was lower, and more pigment stem cells and pigment-producing cells were still in place. The build-up of grey hairs was delayed. They also tried similar plant antioxidants, but they didn't protect pigment stem cells or slow greying in the same way. Luteolin was the one that stood out. For humans, the message is cautious but hopeful, since the same core biology exists in our follicles.